Okay, now from now on, we are going to talk about pharmacokinetics. So this lecture is more about an introduction of what we are going to talk about in the next several lectures. So what is pharmacokinetics? And it's a discipline in pharmaceutical science. That pharmaceutical science is a big category, and underneath it there is a pharmacokinetics. And this discipline studies something can be described by these 5 words. It's a drug absorption, distribution, metabolism, excretion, and toxicity. So we will discuss a lot about drug distribution. So this will be distribution will be like a larger part of the focus of this work, and then we will also talk about other things like metabolism and excretion. So when people talk about the pharmacokinetics, and even in daily life, if you have like shadowing some clinical spaces and other spaces, you might have heard people talk about drug concentration. So let's first talk about what do people mean drug concentration. Our body has different tissues, organs, and different forms of those existence. For example, you have fluid phase. Fluid phase including plasma, interstitial fluid, intracellular fluid. Those are that we have been talking about. And you have like proteins. Proteins can bind to drugs. And there are a lot of like other parts of the body which are not fluid. So given the entire body, what does it mean when people say drug concentration? As you can see, if you try to ask deeper, it becomes vaguer. Do you mean drug concentration in the lung or in the kidney or in the liver or whatever? So here in practical sense, people usually refer to like by default. Okay, let me see by default. Drug concentration refers to the concentration of drug in plasma. Plasma is the liquid part of the body we have been talking about extensively. And this is just by convention. So you know that drug can be anywhere in the body, not literally anywhere. I just means it can travel in much wider spaces other than like besides plasma. So in other places, they may have different concentrations. And usually from clinical experience, people find that if they just refer to drug concentration in the plasma, that will be good enough, like roughly on the bulk part, that people can use this concentration to study how drugs impact on the body. And another practical reason is the concentration of drug in the plasma is something can be easily measured. Someone like can take a blood sample from a patient and then measure its drug concentration. It's so easy. However, if you wanted to know the concentration in other parts of the body, it's not so easily measurable. So, of course, urine secretion that's another category, but we will discuss that later. If people would like to know when the drugs are still in the body, like before being secreted, like before being filtered by kidney, going to the urine, like within the body circulation, like what's the concentration, then people would refer to the concentration in plasma. So that's a notion you needed to know. Okay, so in the plasma or in the blood, or let's say, or still in the plasma, plasma you have liquid phase, liquid, and a protein. If you think about the drug within the plasma, it can just exist in the liquid phase. It can also bind to protein. So when people talk about the concentration of drug in plasma, sometimes they may tell you they refer to the total concentration. Sometimes they may be specific about which concentration they refer to. Let me put it this way. So C total is C T plus C U. So C total is the total concentration of drug in the plasma. And the CP is the concentration of drug that is bound to proteins. Those are the proteins in the plasma. And the Cu is the concentration of unbound drug. Usually, the concentration of unbound drug is what is pharmaceutically important, because those are the drugs like freely moving in the liquid phase, and they may be transported across the capillary wall. We have been talking about capillary membrane permeability that can allow particles to go through, and the drug is one of them. So those unbounded drugs in the liquid are more likely to go through the capillary wall and go to the tissues, go to the cells, and go to where they need. So usually people would like to know Cu. And in order to capture this quantitatively, sometimes people like to define Fu called unbound fraction. So Fu is called unbound fraction. It is defined by the concentration of the bound drug concentration divided by the total drug concentration within the plasma. Under physiological conditions, the unbound fraction for many drugs, they are roughly the same. Like they are not exactly the same, and some drugs may behave a little bit different from others. But like roughly speaking, you can approximate as something you may know, or in clinical setting for a typical drug, people may expect, okay, this unbound fraction may roughly be at some level. So for this reason, if Fu, the fraction, is roughly known, then if people only give you the total concentration, then you know roughly what's the unbound concentration. Or if people just give you the unbound concentration, you can also roughly know what's the total concentration. So that's the a common practice in this field. Now we are interested in knowing drug concentration. And in order to have a better understanding of how plasma drug concentration changes with time, let's look at some curve. So in this type of discussion, pharmacokinetics, we are interested in. So interested in knowing drug concentration. It's a function of time. In dynamics modeling, and in a lot of like biochemical modeling, whenever things are a function of time, people usually call them kinetics. You may have seen this type of terminology in other contexts when you study like physiology or quantitative biology. People still refer to kinetics. So for example, if we want to know concentration C, we want to know its profile. So now this will become our temporal profile. Temporal profile of drug concentration. In the previous lectures, we have been talking a lot about the the spatial profile of drug, for example. Oh, sorry, not the drug. A spatial profile of oxygen or solute, we would say C is a function of X or Y or Z. Maybe they are also a function of time. And in those scenarios, we are concerned about both the temporal and the spatial distribution. And in the discussion of pharmacokinetics, usually people are more interested in the distribution in time. We will discuss later why that's the case in the next lecture when we discuss modeling. So let's look at three typical scenarios of pharmacokinetic curves. So here, the x-axis is C, is the concentration of drug. The y-axis is time. Let's first consider the first scenario. Curve 1. This represents a case which we call the absorption. Example can be like oral sub okay, yes, or transdermal administration of drug. So this absorption means it takes time for drug to absorb in the body. So the t equal to 0 means the time people started to like swallow a pill, and it wouldn't like immediately be measurable in the plasma, and it could go to the stomach and then go to intestine and then go through the circular system and being measurable in the body. So initially it starts with 0 and then starts to increase as it's getting absorbed. And then later on it decreases because job being like eliminated or metabolized. So this curve represented the classical absorption elimination kinetics. So this is the classical absorption, elimination process of kinetics. Okay, now let's look at a second type of curve. This type of curve is referred to rapid intravenous injection. Because it's a intravenous injection, it immediately spikes in plasma concentration. And then when it's getting metabolized or eliminated by renal, and it started to decay. So this is what happens if some patient get an injection, a rapid injection in the venous. Because of this like faster and immediate accumulation of drug in the blood, in the plasma, so people need to be careful of control the dose, because if it's not well controlled, they need to think about, well, how do they avoid the toxicity and other scenarios due to the high peak concentration. The third scenario is this is continuous intravenous infusion. So this is a slow rate of like infusion. So it's administered at a constant rate. So the drug administered at a constant rate. So that the input of the drug, so like if you think about the body as a control volume, or like the factor part of the body control volume, then the influx is what comes in from the intravenous infusion. The outflux is what's being secreted or eliminated through the kidney. So then the in rate and out rate on the steady state, the two rates should be the same. And if the two rates are the same, then people would expect like almost a plateaued or constant concentration of drug as shown in this curve number 3. And once the infusion is removed, then the concentration in the body, the concentration of the drug in the plasma immediately drops. So these are the 3 typical ways that how concentration of drugs evolves with time in the body. Okay. I think that's all the context. Let's just leave a quick exercise. This is a very simple one. Just practice the basic concepts. So a particular drug is known to have a total plasma concentration of a given value, and 85% is bound to plasma proteins. Ask us to determine the unbound drug concentration in the plasma. So this is just a direct use of the definition of unbound drug concentration. All right, that's all for this lecture.